Dr. David Antonetti, University of Michigan, and he will talk about mechanisms of blood to retinal barrier permeability and potential links to angiogenesis.
Well, thank you first to the organizers. A really interesting and exciting format to talk about blood vessels. And it's been particularly, I think, great all day. I've gotten a chance to see people that we normally don't get to see, people I've had a chance to read for many years, and now I get a really chance to interact with. So congratulations, and thank you very much for letting me participate. I'm going to try, obviously, in this very short amount of time to speak broadly and use schematics for a lot of what has been published, and then talk about some new work. So we're particularly interested in the blood-retinal barrier, and particularly as it relates to diabetic retinopathy. And here is what's a fundus photograph of what a clinician would look like with a patient with diabetic retinopathy, and the problem is this central macular edema, which is very closely associated with loss of vision. We have these microhemorrhages, lipid exudates that are occurring. And if you do a fluorescein angiogram on the right here, so just perfusing fluorescein, and it tells you a lot, and you can see a lot of the changes. So we see these blood vessels that leak. We can see this leak at the optic disc. And then we see some of these very interesting regions. We see regions where there's very low perfusion of blood vessels, and beside them, we can see these hyperpermeable regions that are associated with neovascularization. And so some of the questions that we have is how are these blood vessels leaking, and how is this proliferation occurring? Of course, I'm sure everybody here knows vascular endothelial growth factor, or VEGF, is an elevated factor in this disease process. And by delivering anti-VEGF therapies into the vitreous of the eye, about half of patients are showing a good responses, and some even improving in vision, and many having less loss of vision. So we can reduce the permeability in clinical studies looking at this angiogenesis. So, a lot of the perspective so far, and a lot of what I'm going to talk about today, has been how do we block the pathology moving forward. And in a nutshell, the question that I would have going forward for our group is, can we, as we start going forward, thinking about changing that and asking, instead of blocking this neovascularization, why is this pathologic neovascularization rather than a reparative vascularization? And how could we potentially promote a reparative vascularization? So I just basically said that. This is a schematic from a relatively recent review from Harris and Nelson, and Dr. Malik very beautifully talked about a number of these parameters. This is looking at adherence junctions, and this makes a really important point. So Dr. Malik talked very much about this side of the picture, which we see lots of permeability factors that signal through many of the well-studied events have been through kinases, tyrosine kinases, as well as serine kinases that can phosphorylate proteins such as VE-cadherins or related catenins, and these are associated with permeabilizing events. Endocytosis appears to be a major part of this process. So that's going on. But this side of the picture shows another important point, which is that this adherence junction complex then impacts the endothelial cell and is also a signaling center in that endothelial cell. And so VE-cadherin can downregulate, in a complex, can downregulate VEGF receptor signaling. These adherent complexes can regulate cytoskeletal organization through G proteins, beta-catenin, or VE-cadherin through FOXO can regulate tight junction, particularly Claudin, like Claudin-3 expression, and Claudin-5 expression, I'm sorry. And beta-catenin can regulate Claudin-3 expression, regulating barrier formation. So we have signaling going to the adherence junction, regulating permeability, but also the adherence junction can act as a signaling center, affecting things like cytoskeletal regulation and junction formation. We'd like to argue that the tight junction complex also plays a similar role in addition to the adherence junctions, particularly in tissues like the blood-brain and blood-retinal barrier. So there's lots of data that suggests that occludin cloudins are altered in expressions. A lot of this data comes from epithelial cells in cancers. Very interesting data about ZO1, a junctional protein that moves to the nucleus, and ZO also being associated with growth control, specifically through binding proteins such as ZONAB and CDKs, and ZO2 also binding other regulatory proteins in growth control such as Jun and FOS. Our favorite tight junction protein that we spend a lot of time on is occludin. It's probably a misnomer. It probably isn't really involved in creating a barrier. Knockouts have been made and animals live, but they have a host of defects. And these defects include some cells, mucous cell hyperplasia. Occludin has also been shown to target TGF-beta receptor to tight junctions, be required for epithelial to mesenchymal transition. As I said, it's lost in a host of epithelial cancers. And very interesting studies showed transformed PA4 cells can be reverted by overexpression of occludin. And we've shown in ARP19 cells, siRNA to occludin also leads to proliferation. So a number of years ago, our lab and a number of other labs had identified it, started working to identify post-translational modifications, specifically phosphorylation sites. In red are serine-threonine phosphorylations, and blue are tyrosine phosphorylations. And we're just learning about how all these phosphorylation events lead to regulation of cellular function. And what many of us are coming to is that occludin is not really occluding at the tight junction, but a regulator of barrier properties and has a variety of signaling processes in the cell. So just to point out a few, serine 408 was studied by Jerry Turner's lab and was critical in forming a cloud and pore, which allows permeability very specifically to ion flux. And one of the things that, from our perspective as a tight junction biology lab, we've noticed today is that we talked about permeability in general, but permeability is very specific. There are different routes for different kinds of molecules, and this was actually a very good example of a very specific pore formation allowing ions that wouldn't allow large solute flux. Tyrosine 471 looks to be pro-barrier. Phosphorylation looks to be required for assembly of the junction complex, and that's an area of ongoing work. This talk today, we're going to focus on serine 490 phosphorylation. It's in this region of the coiled coil terminal domain of occludin, and it's right on one of these turns. It's a PKC target, much like we know PKC alpha targeted V cadherin for permeability control. This also appears to be a classical PKC target. So this is a rather complex slide, but I just highlight a few points. This is endothelial cell culture looking at permeability in response to VEGF, and we see our typical twofold response, and this is looking at large solute flux, 70 kilodalton dextran. If we put in PKC beta, we get an even larger increase in response to VEGF. And if we overexpress a point mutant at serine 490A of occludin, we can completely block that VEGF effect, even if we're overexpressing PKC. Conversely, if continuing work showed that this phosphorylation led to a subsequent ubiquitination and endocytosis of occludin. Very much like V cadherin undergoes endocytosis, we think both processes are occurring in parallel. And if we put in an occludin ubiquitin chimera, we could make the cells constantly leaky, even if we had a dominant negative PKC present. So this led to a model in which we believe occludin is phosphorylated in a PKC-dependent manner, leads to ubiquitination and internalization, in fact, able to show co-precipitation with a number of these ubiquitin interacting motif-carrying proteins that act as chaperones, and endocytosis of occludin and other junctional complex. But we had an interesting surprise, and VEGF, of course, is not only a permeabilizing factor, a proliferating factor for endothelial cells. And with our phosphospecific antibody, we would notice cells with condensed nuclei that were actually going into mitosis. You can see here the centrosome for gamma tubulin, and we can see very clear staining for the phosphooccludin at the centrosomes in these cells as well. And all through mitosis, metaphase, anaphase, and telophase, you can see really very nice co-localization with gamma tubulin. If we transfected then endothelial cells here with the GFP tag, we could show the GFP tag co-localizing very beautifully also with the centrosomes, and here the anti-GFP antibody around this condensed nuclei. So it wasn't the antibody binding something else, it really was occludin. On the left is a patient that had to undergo an occlusion for proliferative diabetic retinopathy with an epiretinal membrane. So here, this is this neovascularization growing into the vitreous. It was surgically removed. And from that surgical sample, we stained and were able to see phosphooccludin that perfectly co-localized with centrosomes as well. So we had this unique localization for this tight junction protein. We looked to explore this more, used the tube formation assay. So these are endothelial cells in culture and inducing tube formation with VEGF. And so you see over time, 24 hours, this tube formation. And over time, we see this increasing occludin serine 490 phosphorylation. So does this occludin phosphorylation significant to the process. So if we transfect cells again with this 498 point mutant, the same point mutant that blocked VEGF-induced permeability, you can see we were able to completely block this tube formation in response to VEGF. So here's wild-type occludin. It allowed tube formation just fine. The 490D mutant allowed, phosphomimic allowed tube formation, but the 490A completely blocked tube formation. That's shown quantitatively here. It also blocked endothelial proliferation, so using incorporation of either DUTP or other methods, we can have the VEGF-induced proliferation. Wild-type occludin actually showed a slight reduction, and the 490A completely blocked the VEGF-induced DNA synthesis. Conversely, if we knock down occludin in these endothelial cells, and this is knocked down here, we see a basal increase in proliferation, a basal increase in tube formation, and an increase in VEGF-induced tube formation as well. So it appears that occludin puts a block on the proliferation and tube formation that may be released by phosphorylation and potentially in a PKC-dependent manner. Looking in vivo, we used a model developed by Peter Campachero in which VEGF is expressed in an inducible manner from the outer retina. And so we can induce this, and when we do that, we cause the vasculature to grow down into the outer retina. It's so dramatic, the retina actually will come off in about 4 days. If we look at that, this is a picture, just an IB4 staining cross-section of the retina. We see this increased vascularization occurring. And if we look at this serine 490 phosphorylation, we see again the dramatic increase in phosphorylation quantified here. So what are these vessels like? And they are hyperleaky, just like we might expect with a pathologic neovascularization. So I'm not going to describe this because I don't have a lot of time, but the NHS biotin assay to identify regions of leak. And if we're looking at the deep capillary layer, this is in the control, it's very little leak, but after the VEGF induction, we see a very dramatic leak. And if we look at the occludin localization in this deep capillary and the ZO and the control, it's nicely organized. And after DOC's induction of the VEGF, that occludin is really, really lost in its organization as well as ZO1. So finally, can we reverse this in vivo? We worked with Dr. Bill Hausworth, who has a AAV virus that is able to transduce specifically endothelial cells, and we were able to do this in the eye of mice, and we use this VEGF induction model. So if we just put in GFP without DOCs, we see the IB4 staining of blood vessels right where they're supposed to be. If we put in the GFP and induce the VEGF expression, we get lots of angiogenesis, pathologic angiogenesis. The wild type occludin did not block that pathologic angiogenesis, but transduction with the S490A point mutant of occludin really blocked that pathological angiogenesis or neovascularization. And quantified, of course, in a mass fashion, we were able to show that we really completely blocked that, that neovascularization in vivo. So we think that in a short time frame, VEGF induces an occludin phosphorylation, we think at the junctional border, that's associated with endocytosis in a pathway that's probably very much parallel, maybe likely integrated with VE-cadherine endocytosis, and that creates breaks and permeability, large permeability pores that would allow things like dextrans and albumin to flow through and increasing permeability. This is maybe a 30-minute time frame. And then after a 12 to 24-hour time frame, we see again an even stronger phosphorylation of occludin. Here we see it very much associated at the centrosome, and this is associated with proliferating cells and appears to be a required step. We don't know exactly how yet, but it may well be associated with orienting the cell. An effective angiogenesis, especially with cells that have these junctional complexes, really may need to maintain an orientation, and we think that that might be what's involved here. To be very precise and brief, our long-term goal then is, we've been blocking this process. We'd really like to know how we could go from here back to a quiescent vessel. So how can we promote a reparative system? So lessons learned, the junction complex may be regulated by trace translational modifications to provide a specific alterations in permeability. And formation regulation of the junction complex is intimately linked to growth and differentiation, and junctional complexes may regulate proliferation, migration, and angiogenesis, not just from our work, but from a host of other laboratories that have been showing both occlusion, ZOs are involved in regulating the growth process as well as the barrier process. And as we said, gaps in our knowledge, where can we go? Can we think more about understanding reparative angiogenesis? And I thought Dr. Davis brought this up first thing in the morning. I couldn't agree more. Potentially by studying normal developmental angiogenesis and comparative analysis to pathologic angiogenesis, we can identify important differences and learn how to promote angiogenesis that could be effective. And the last point, I didn't get a chance to show data or slides, but with our keynote seminar from Dr. Aird, we completely agree. We see very interesting differences between the different vascular beds. For example, this occlusion phosphorylation seems to be very much restricted to capillaries, particularly in ischemic reperfusion in eyes, while we see very different changes, but very important changes in the junctional complex and large vessels. And so there appears to be very important differences that are not going to be apparent by smashing up the whole tissue. Further understanding the specificity of the vessel changes in response to these pathologies, I think, is going to help our understanding greatly. Thank you very much.
Thank you. We'll take some questions.
Dave, really nice work. Congratulations.
Thank you.
Just a comment and then a question. So there's good precedent to indicate that post-translational modification of junctional components, i.e. a breakdown of the junction via proteolysis, is a means by which you can activate transcription, for example, in Notch or in catenin signaling, where pieces of the junctional components actually transmigrate to the nuclear compartment and actually stimulate mitosis or other gene transcriptional events. Do you know, have you looked, is the VEGF-dependent phosphorylation on occlusion at S490, is that representative of the intact molecule, or is that actually a piece of the molecule?
Great question. I'll just start by saying, we've done some evolutionary analysis of the molecule, and really occlusion looks to be a joining of two parts. That transmembrane is in fact, it's part of a family of a MARVL, and MARVL D3 is just the transmembrane part. And that long cytoplasmic tail and the coiled coil looks like it's also called the L domain, and it looks like it's derived from another, and it looks like it was a fusion vet over time. We were sure that the coiled coil was going to be cleaved and moved to the centrosome, but we did do a centrosome prep, and we do see the whole occlusion at the centrosome. So, yeah, we're still trying to understand. There are some other transmembrane proteins at centrosome, so we're not out there alone, and we're trying to understand that.
Thanks very much.
Yeah.
Thank you. Next gentleman over there.
So is it a common feature of the process of the choriocapillaries versus the retinal blood vessels?
Oh, that's a great question. We don't have an answer. So far this has been focused on cells that express a high degree of tight junctions and occlusion. I don't know. I think that's a great question. We haven't done anything to ask the question about the choriocapillaris, which is just below the retina and supplies all the outer retina, and part of AMD is the angiogenesis coming up. We have no idea if occlusion plays a role in that vessel bed or not.
But after angiogenesis, could they maintain the junction? What is the nature? Because most of the neoangiogenesis process make leaky vessels. So these junctions might be very leaky.
I completely agree. We think these junctions are very leaky, and I think one of our really important questions is to understand why in development that doesn't happen, and why in these pathologic conditions this is very different kinds of angiogenesis.
Okay, we need to move on. Next question, please.
Very nice talk.
Thank you.
My question is about the tight junction that induced by the VEGF signaling towards contact inhibition of endothelial cells, and whether this is conflict with this potentially growth inhibitory factor for you. You see the growth promotion effect.
Do I think it's different? I don't know.
Do you think contact inhibition is different?
In our hands, endothelial cells have some degree of contact inhibition, but it's not nearly as powerful as epithelial cells and epithelial cell cultures that can really shut down really tightly with a contact inhibition. But I'm not sure if I get the essence of your question.
Yeah,
maybe we could talk after. Or should talk after.
Contact inhibition, we will usually interpreting contact with the growth inhibitory factor of the growth endothelial cells, which will be anti-angiogenesis. angiogenic.
Oh,
so
sure. And
there will be conflict with your theory.
absolutely. And a recent beautiful modeling paper about angiogenesis showed that the cells in the angiogenic tube have to keep moving, and that junctional complex has to be released and allow those cells to move, and that's an important aspect of angiogenesis. The centrosomal localization we think is important, but the release of the junction appears to be a central part of angiogenesis as well. Yeah.
Next question, please.
Great presentation.
Thank you.
So, occluding knockout mice have brain calcifications.
Yes.
And there is evidence that some patients who have a loss of function, occluding mutations, also have basal ganglia calcifications. So the question is this a problem just with extravasation of blood leading to secondary, inflammation, and then ends up being into, or there is more of an active matrix remodeling process that affects that?
Yeah, I don't know the answer to that question. I think the brain calcification, and I remember when Dr. Ferrucci first presented, they really didn't have a handle on why that's occurring. I think those are great possibilities, and I don't think we have I think we're just kind of getting to the point now where we're starting to understand that this is not a central barrier, but a regulator of the barrier and how it might do that. So, yeah.
Next question, please.
Yeah, I didn't see any cells in your retinal preparations, but is this a cellular process, or is the VEGF produced by some other component and then leads to a leaky vessel, or is it actually an inflammatory type process?
In the in vivo system that we had?
The one that you had the fluorescein leaking from.
Yeah. Okay, so it's a system in which it's an inducible VEGF from the rods. So it's an artificial system. It represents a specific type of AMD with retinal angiogenesis. Normally AMD would be from the choroid. This is a very specific subtype, but it's an excellent model because the outer retina is normally avascular. So when we induce that VEGF from the rod, we induce those endothelial cells to grow down into the normally avascular retina. So we were just using that system.
If you did that in a patient, would you see inflammation, or would you just see?
So I think that's a great question. We are just starting to ask the question of the relationship between the inflammation. I will tell you this real briefly. We've injected VEGF into the eye. We get very little inflammation. TNF very little. We co-inject VEGF and TNF into the vitreous, and we get a bomb of inflammation.
Thank you.
Thank you. Last question.
Thanks. So I'm just curious, one of the things that been missing, maybe just because I'm interested in calcium, calcium signaling is obviously very important in endothelial cells and can dynamically regulate at least their retraction, or maybe the endothelial stiffness as well. How would you envision this fitting in? Because this potentially is a mechanism to rapidly change endothelial cell morphology. It's been missing from all the slides I've seen so far.
We think about calcium in terms of, right now, the classical signaling of PKC as a required element for the PKC signaling.
Non-muscle myosin?
I guess I'm not going to, probably wouldn't be best for me not to comment on that.
So maybe I can follow up. There's
Yeah.
actually a really important role that calcium plays in regulating the remodeling of the endothelial cytoskeleton through the work of calpain. M-calpain and mu-calpain are activated locally by calcium, which goes on to cleave cytoskeletal and junctional components that foster the remodeling, stimulate the migration, and induce the proliferation.
Okay, so